Değerli hocalarım, sevgili katılımcılar, 2. Multidisipliner Sinirbilim Kongremiz kaldığı yerden devam ediyor. Ben bu oturumdaki moderatör Bade Buse Bakır. Bu oturumumuzun sonunda çekilişimiz olacaktır. Ve çekiliş formumuz oturum boyunca açık olacaktır. Çekiliş formuna açıklama kısmından ulaşabilirsiniz. Soru cevap kısmı başladığı anda çekiliş formumuz kapanacaktır. Sorularınızı soru formumuza doldurarak hocamıza iletebilirsiniz. Ben sorularınızı ileteceğim. Sorularınızı İngilizce olarak sormanızı rica ediyorum. İngilizce sorulara öncelik verilecek. Ve bugünün 5. oturumundayız. 5. oturumda Bilkent Üniversitesi Psikoloji Bölümünden Aysel Sabuncu Beyin Araştırma Merkezi'nin kurucusu Profesör Dr. Michelle Adams, Brain Aging, Determinants that could lead to or prevent cognitive decline adlı sunumuyla sıradaki konuşmacımızdır. Now, Professor, I will turn my camera and sound off, and we will begin. Thank you.
Okay, great. Thank you for that introduction. Can you hear me okay?
Yes, absolutely.
Okay, all right. So I want to thank the organizers for allowing me to come and inviting me to come and give a talk today, which will be about brain aging and the determinants that can lead to or prevent cognitive decline. So we all know that as people get older, many cognitive domains change in our abilities to perform those cognitive behaviors. And one of the most significant changes that happens with aging are changes in learning and memory ability. And this is something that is can be varied in terms of its spectrum, and it's also difficult to cope with and for older adults. In the laboratory, we can actually measure these changes on a virtual behavioral task. This is a virtual Morris water maze task. And in this task, the subjects need to find the location of a hidden platform. And you can see it in the picture here. It's, you can see it, but normally it's hidden under the water. And they use the cues around the maze to locate this platform. And basically, it's similar to a task you would use to navigate around your house or around your university campus. So it involves spatial learning and memory to bring up that spatial map. And this task is actually based on a task that was developed in rodents. And what you'll see is that young subjects perform well on this task in most cases. Some are not as good as others. So there is a variability in young. But what you'll really notice is that older adults have a lot of difficulty with this task. So finding a location of this platform becomes harder as one ages, although some still perform equally as well as young subjects. And then what we can do is in this task, we can actually measure the subject's memory for the location of this platform. So what you'll see here on the left is a young subject, and on the right is an older subject. And in this case, normally during the training phase, once the subject enters into the quadrant where the platform is located, it'll actually pop up. So it gives them a reinforcement that they know they're in the right location. In this case, when we measure the memory for this platform, we actually keep it hidden and see if the subject will continue to stay in that location because they know that that platform should be there even though it's not coming up. And in the on the older subject, what you'll notice is that not only do they enter into the quadrant where the platform should be, but they're also swimming around and in some cases staying longer in places where they shouldn't be. So they have lost their ability to recall very precisely the location of this platform. And this is actually performance across different training trials. This is where the subject enters into the quadrant and the platform comes up. And what you'll notice is that both young here is in the diamonds and the older in the squares, both groups improve across time, but older individuals are significantly worse than younger individuals, although there's some variability. And what they've also determined is that if you look for the biological substrate of these changes, that they've actually looked at the volume of the hippocampus. And that's this green structure right here. It's bilaterally located in the medial temporal lobe. The reason being is that in animal models, if you lesion this hippocampus, so you take out the hippocampus, animals are impaired on this type of task. And what they've shown in aged subjects, so the aged individuals are in the open squares and the young subjects are in the closed black circles, is that the time spent in that platform quadrant, meaning the quadrant where the platform should be located, correlates with the size of the hippocampus. So if the subject is spending less time in the quadrant, they actually have a smaller hippocampal volume than subjects who are spending more time in the quadrant, and they have a larger hippocampal volume. So the size of the hippocampus relates to a subject's performance on this task. So what could a change in size mean? Well, it could mean a lot of different things at a cellular level. And one of the earliest thoughts is that it reflects changes in the numbers of neurons in the brain, and this is going to contribute to the size difference that they've seen with the MRI. And what is becoming very clear is that there is actually no significant neuron loss in the aged brain that's happening across normal aging. So this is a separation from what we would see in a disease process, like a neurodegenerative disease process. And this is actually two different studies, one that was done earlier and the one was done later. And this was actually done with a little bit less precise counting methods. But in both cases, they actually show that there's no significant neuron loss. There's variability in how much is there. Also, gender can play a role in this, but significant neuron loss is not happening in the aged brain. So this actually leads us to the question about what are the cellular molecular changes or signatures that are happening in the aged brain that could be contributing to these changes in cognitive function. And to understand this, my laboratory has been using both rodent and zebrafish animal models to be able to get down to the specifics of what's happening at a cellular level. And we're particularly interested in this hippocampal structure because of its relationship to the spatial learning and memory changes that you see in the behavior. So we know, I told you that this virtual Morse automaze task is actually a task that was developed in rodent models. So both mice and rats have been tested in their behavior in this task. And we know, just like humans, rodents, when they're actually trained on this task, are significantly worse than young subjects. So you actually see a similar change with aging that you do in humans. And we also know that zebrafish can show similar age-related changes, and that, more importantly, they actually have a similar structure to rodents that is equivalent of the rodent hippocampus. And this is known as the pallium, and particularly the lateral pallium. And what they've shown in zebrafish is that when there are lesions in this dorsal telencephalon area, the pallium, they actually are disrupted on a spatial memory task in a similar way that in young subjects, young rodents, when you lesion the hippocampus, they're disrupted on a Morse automaze task. So both zebrafish and rodents have a similar structure to the human hippocampus. Rodents, it's hippocampus, and zebrafish, it's pallium, that performs a similar behavioral, controls a similar behavior, which is spatial learning and memory. More importantly, we also know in zebrafish that they, just like rodents and humans, show changes in age-related decline. So if you test them on a spatial memory task, they actually show decreases between young and middle age and young and old age. And in this case, young is actually 1 year, middle age is 2 years, and old is actually 3 years. On average, zebrafish live 3 to 5 years. that's a little bit longer than rodents, but in terms of when they're showing spatial learning and memory changes with age, it's actually very similar. So this is an important information to obtain, because we need to know that zebrafish have a similar nervous system and structure, in terms of structure and function, to rodents, to be able to use this model to understand changes that are happening in human subjects. So, do they exhibit significant neuron loss? I've told you that in humans, no neuron loss is happening with age, and actually what is becoming very clear is that in the rodent brain, significant neuron loss is not happening during aging. So in this particular study, the authors took young and old rats. They behaviorally characterized them on the Morse-Water maze so that there's a group that is unimpaired on this task and a group that's impaired. Because if you remember, there's variability even within young, but especially within age groups, how well somebody is aging successfully versus unsuccessfully in terms of cognitive impairment. And then counted the neuron number in every subfield of the hippocampus, so dentate, gyrus, CA3, and CA1, and found no significant difference, even when the animals were separated into how successfully they're aging in terms of their cognitive abilities. Then they looked actually for correlations between neuron number and performance on this task, and saw no significant relationship between the number of neurons and how well the animals performed on this task. So again, just like humans, there's no neuron loss in the rodent brain that's contributing to changes in age-related cognitive function. Now, my laboratory actually took young and old zebrafish, and we counted neuron number in the lateral pallium, or in the pallial structure, which is the homologous of the rodent hippocampus. And we concluded both males and females in the study. So the rodents data I showed you was only males, because we also know there's some variability that happens to whether that subject is male or female. And for recording these or counting these neuron number, we actually used markers of neural stem cell or neural progenitor cells, so this double cortin-like kinase, QC, which is an immature neuronal marker, NUIN, and MAP2, which are both mature neuronal markers. And just like rodents, when you look at young versus old, although there is variability in neuron number, there's actually no significant neuron loss that's occurring in the zebrafish brain. So both rodents and zebrafish show similar cognitive changes with age, and they show similar changes in neuronal numbers, in the fact that there is no significant neuron loss that's occurring in the brain. So if there's no significant synapse loss, or neuronal loss, sorry, what are the neuronal changes that may be contributing to these changes in cognitive decline? We hypothesized, as well as others, that there are subtle but significant changes in the cellular and synaptic function and regulation. So it's something that's happening within the cells, or within the cells in terms of their structure or their function, how they're functioning with age, that are contributing to these cognitive changes. Unlike a neurodegenerative process where you're having actually cells die and degenerate, you're actually seeing changes in the subtle change in the cells and the synapses. So what are some of the targets that you can actually look at in this case? Well, in terms of cellular targets, you can actually ask questions about whether you're looking at glial changes, so these are supporting cells, or are there changes in the regulation of these neuronal markers, these proliferative progenitor markers, these immature markers, or these mature markers. And then in terms of the synaptic changes, you can actually ask questions about number of synapses. You can actually ask questions about size of synapses. You could actually ask questions about whether you're looking at excitatory versus inhibitory type synapses. And then within these excitatory synapses, there are proteins that you can actually ask, are those protein levels changing? So there are presynaptic markers such as synaptophysin, which is a presynaptic vesicle marker. We can look at postsynaptic markers that are related to function. AMPA and MDA are the major excitatory neurotransmitters channels that are contributing to synaptic function, as well as their clustering proteins. So there are postsynaptic density 95 and different associated proteins. Within the inhibitory synapses, again, synaptophysin is found there because it's a general presynaptic vesicle marker, but GABA receptors are, GABA A in particular, are the major excitatory or inhibitory channels that are going to mediate fast inhibitory transmission. So there's lots of targets, and by identifying these targets that change with age, you can understand whether there's a potential for drug development to modulate their function and change what's happening in the age brain. So we know that in terms of function, there's also changes in cells that is happening with age. So synaptic plasticity, which is measured by long-term potentiation and thought to be a cellular basis of learning and memory, is altered with age. So in this case, these are young animals here with the closed black circles. They're old animals that have been successfully aging in terms of cognitive abilities. And these are rats, by the way, male rats. And then we have animals who are not successful aging in terms of cognitive changes. So those are the open circles. And what you'll notice is that the long-term potentiation actually has a faster rate of decay in aged animals. Their magnitude is actually decreased. And the amount of synaptic input, so the threshold for inducing this, actually changes with age. So functionally, synaptic plasticity is changing. Now, in particular, people tend to associate changes in long-term potentiation, or LTP, mainly with changes in excitatory function, but it could also reflect changes in inhibitory function if it's overall at a system level, thinking about how much excitatory versus inhibitory balance is changing, that would affect how long-term potentiation is manifested in the age brain. So initially what we asked is, what's the progression of glutamate receptor decline across lifespan? Because we wanted to initially look at excitatory type of synaptic changes. And can we alter the course of glutamate receptor changes by diet? Now, when we study aging, it's not enough just to study what's happening with age. We also want to study potential interventions that might alter the course. Because when you study an intervention, you can actually then go on to identify potential targets, modulation, and maybe whatever intervention you're applying, whether it's actually some sort of environmental or lifestyle change versus a pharmacological change, this would actually help to then have a more translational potential to human populations. So the dietary manipulation we did was caloric restriction. So this is the only non-genetic intervention that's known to reliably increase health span and lifespan. And this caloric restriction is different from starvation. So caloric restriction is basically a daily reduction in caloric intake across a person's, for a certain period of time. And typically it's been induced across lifespan. So basically from the time the subjects are sexually mature until end of life, they are raised on this calorically restricted diet. And it's across multiple species, monkeys, worms, rats, it actually increases both the health span and the lifespan. And then there are actually groups of human subjects that are doing this voluntarily. And when we're talking about the type of caloric restriction that we're giving, it's a daily reduction to 60%. So it's a 40% reduction in total caloric intake, which is quite severe. So if we can find pharmacological ways to mimic this caloric restriction, so a caloric restriction mimetic, it would actually be better for people. So when we apply this, again, these are young, middle-aged, and old male rats. And from the time they were 4 months of age until the end of life, they've been on this restricted diet. And the young animals were 12 months old at the time of the measurements were taken. Middle-aged were 18 months, and the old were 28 to 32 months. What you'll see is that normally from young to middle-aged and old, there's a significant increase in body weight that happens with aging. And when animals are calorically restricted, they actually lose 40% of their body weight, and it stabilizes across lifespan. So there's no significant change in body weight once an animal is calorically restricted from the time beginning of the following sexual maturity until end of life. And so this is consistent with lots of literature. Then we actually wanted to look at one of the factors that they think is contributing to the increased health span and lifespan, and that's changes in insulin-like growth factor 1. So what they believe is that by calorically restricting animals, you're actually lowering the levels of insulin-like growth factor 1, or IGF-1, and this lowering actually is beneficial to the health span and lifespan of the animal. Because normally, actually, between young, middle-aged, and old, you actually see a significant loss. Although it's small, it's significant. And in calorically restricted animals, just is following a similar pattern to the body weight, where it's significantly reducing but stabilizing across lifespan. So the idea is that by calorically restricting animals, you're actually changing the levels of this insulin-like growth factor 1. And by changing it, not only, it's not so much the absolute direction it's happening, the change, but the fact that it's actually stabilizing is what is thought to be a benefit of the caloric restriction. We also looked at the changes in behavior. So we took these young, middle-aged, and old animals, and we tested their performance on this Morris water maze test. And what you'll notice is that ad libitum fed versus calorically restricted young are not different in their performance. Middle-aged is also not different in their performance. And you still see a slight decline into middle-aged. But what happens is that this diet actually stabilizes the performance into old age, and it in animals that are actually allowed to eat as much as they want, they're ad libitum fed, they continue to get worse. So this caloric restriction diet is actually stabilizing performance on this task, so that by the time an animal gets older, animals that have had this dietary restriction actually are doing better than those that are continuing to eat as much as they want. So it has a beneficial effect in this regard, and this is actually also similar to what's been described in the literature. So we then took the hippocampus and we actually measured the levels of the glutamate receptor ion channels. We did this in a subunit specific manner. So we looked at each individual subunit of the NMDA receptors and each individual subunit of the AMPA receptors. We looked at a presynaptic vesicle protein, synaptophysin, and we had actin as our loading control. And then we quantified the levels of these in a subunit specific manner. Now, the data here are shown in this slide. So when you look at synaptic proteins, and I'm just showing you in one area of the hippocampus, the basic pattern is the following, that animals that have ad libitum fed, if you look across lifespan, the levels of these proteins continue to decline. Whereas those that are actually calorically restricted, they may be a little less than the ad libitum fed counterparts, but between young and old, they actually don't decline any further. So basically the levels are stabilized in old animals compared to young. And what this means is that when you look at the old animals, where you're actually seeing these behavioral differences, if you remember, old animals that are fed ad libitum are worse than old animals that are calorically restricted, the levels of these synaptic proteins, which are going to be mediating fast excitatory transmission, actually follow a similar pattern to behavior, in that those that have a higher level of synaptic proteins are better in their Morris-Watermaze performance than those that are worse. So this diet is stabilizing a level of synaptic proteins, and likely contributing to the behavioral or underlying the behavioral change that we're seeing. So I've just summarized that. Basically, CR stabilizes synaptic protein levels as well as cognitive performance on hippocampal dependent tasks that declines with age. The biggest changes that we saw were actually observed in the CA3 region, which is one subregion of the hippocampus. And it was exciting for us because this was the first data to describe or demonstrate regional effects of caloric restriction on excitatory connectivity in the hippocampus. So I told you that we did this in a subfield specific manner. So we looked at dentate gyrus of the hippocampus, CA3 versus CA1, and the biggest effects we saw were in CA3. And CA3 is actually kind of an ignored region of the hippocampus, but an important one nonetheless. So it's a region of the hippocampus that actually receives convergence of input. So it's set up to actually kind of monitor activity that's happening in the cortex. It also monitors what's happening in dentate gyrus. And it also is monitoring what's happening in itself. So it has a recurrent connection back on itself. So it's computationally is actually receiving all these inputs, so it can detect, are there changes in one area versus another, and maybe this computational thing that it's doing is actually helpful in terms of not, if it's so computationally good, it'll actually be beneficial still in the behavior. It also has been shown to be involved in spatial learning and memory. So selective lesions of CA3 will disrupt spatial learning and memory. And some recent work has demonstrated that NMDA receptor levels, if you knock them out in this region, that animals will have trouble on this task. So it's an important region of the hippocampus. So we then asked, is the synaptic structure in CA3 altered with age and or changes related to diet? So for this analysis, we took out the CA3 region, and we did a synaptic analysis in the most distal part of the CA3 region. And the reason we did this is that this region actually has been shown to lose synapses, specifically in animals that are impaired on Morris-Watermaze test. So when we picked a very important area and with a very specific hypothesis that we thought this region has to be the one that's changing with age. We used electron microscopy to count the number of excitatory versus inhibitory synapses, as well as determine whether there are changes in the spine, which are the structures that are receiving these excitatory synapses. We did a very specific question, and we used young AL versus old AL versus old CR, so ad libitum young versus old ad libitum versus old CR. And when we analyze this, we learned that axospinous synapses decrease with age and don't change with diet. When you look at so these are excitatory synapses. The axodendritic, although numerically they seem to change with age, didn't significantly change with age, but the pattern numerically was reversed to axospinous synapses, and we didn't see any change in the surface area of these spines. So the synapse number, but not size, declines in this area with age and is not reversed by diet. So together we came up with the conclusion that in particular in CA3, the synapse number, but not size, declines with age and is not prevented by caloric restriction. alterations in synapse number and changes in synaptic protein levels are likely to affect the plasticity and hippocampal function. And then CR, that caloric restriction, helps preserve synaptic plasticity in the CA3 region by preventing an age-related decline in synaptic protein levels in the face of synaptic loss. So currently, I actually changed the focus of my group, both in the questions as well as the animal model, because we wanted to ask things like, what are the effects of timing and duration of CR? Everything that I've talked to you about is lifelong. So can we actually see similar effects using short-term or later in life intervention? What are the effects of gender in terms of response to CR? If you look at the neuron number in humans, there's differences between males and females. So this is actually something important, and majority of work is done in male subjects. What are some of the molecular mechanisms through which CR is acting? Again, because we can find the targets, we can actually develop pharmacological interventions. Can we accelerate the aging process in a younger animal? So is the age-related changes we're seeing only due to the fact that we had the organism aging as a whole, or can we actually just change the way the brain is actually responding in the young animal? And can we find and test mimetics to replace the dietary regimen? Because people will be more likely to take a drug than to do this kind of intervention. So for these questions, we use the Zebrafish model, but initially we also had to ask, what are the age-related biological changes in the zebrafish brain, and do they vary with gender? Because previously, the zebrafish model was mainly used for developmental studies because of its external fertilization that happens and the large number of embryos it provides. But we wanted to actually take it into an adult model. So for these studies, actually my PhD student Ilgam, my former postdoctoral fellow Ayça, and my current PhD student Elif actually pushed these forward to be able to apply interventions later. So for the first study, this was done by Ilgam. We took young and old, male and female zebrafish. We actually took their brains out. I already showed you the analysis she did of neuron number, but she also had a set of animals where she took out the whole brain, she isolated the protein, and then performed Western blot analysis on these similar neuronal markers to determine whether there was some sort of regulation of the levels of these proteins in the aged brain. So she did Western blotting for the progenitor marker double cortin-like kinase, immature neuronal marker QC, NUN, which is a mature neuronal marker. And then she had young and old animals, and she had both female and male animals. And what she noticed is that there were gender-related changes that happened with aging. So it was specifically in male animals. So young males actually decreased the level of this progenitor marker, with females not showing any changes with age, and as well as this immature neuronal marker. The mature neuronal marker didn't change in terms of protein levels, and all of this was occurring in the context of no changes in total neuronal numbers. So these neuronal protein levels are varying both with age and with gender. So it's actually important for us to be able to apply to translational studies in humans to understand that there could be differences depending on the gender of the subject, or the sex of the subject. So to get a sort of more global picture of what's happening in the aged brain, to identify the important dependent markers that we could be looking at, Ayça took young and old male and female fish. She dissected their brains, she lysed the whole brain, isolated the RNA, and did a microarray analysis to be able to fish through and really understand a lot of different changes that could be happening, and then validated those findings with quantitative PCR. And what we found is that between young and old, there were about 700 differentially expressed genes. there were differentially expressed genes that were happening just between male and female, and there was an intersection of about 200 genes that were susceptible to both age and gender. And in terms of understanding the subjects, with what these genes were involved, or I don't want to say subject, but the terms or related to what these genes were doing. So these related to things like cell differentiation, neurogenesis, neuronal differentiation. So it was there were very specific and exciting areas that we thought about in terms of neuron number or neuronal regulation that we could look at then when we apply different interventions. So Edith actually wanted to take a different approach, and she actually did a study of young middle-aged and old. So she included middle-aged animals, and this is actually something that's important to do if possible, because sometimes the trajectory doesn't always just keep declining with age, but you might see something specific that's changing in middle age versus old that could be an important time point for applying dietary interventions, for example. And she took out the brains, dissected them, isolated protein, and did Western blot analysis for different key synaptic proteins. So she looked at the NMDA receptor clustering protein postsynaptic density 95. So this is going to regulate excitatory synaptic function. She looked at the presynaptic vesicle marker synaptophysin and the gefrin, which is a clustering protein for inhibitory neurons. And when she took both female and male animals and saw different patterns depending on the gender of the animal. So in young animals, actually males have higher levels of postsynaptic density 95. And females, actually, this level of this protein actually increases with age and no changes in males. When you look at the presynaptic protein, synaptophysin, it's actually decreasing in males. In middle age, you see higher levels than females. And females actually don't show any significant change with age. So males are showing changes in a presynaptic marker, and females are showing age-related changes in postsynaptic marker, excitatory postsynaptic marker. When we looked at inhibitory marker, gefrin, there was actually no change. Numerically, it looked like there could have been some peak in middle age in males, but statistics showed us there was no change. And when you look at our internal protein control, tubulin, it's actually not changing with age. So this was our housekeeping protein. So these synaptic proteins are changing based on age and gender in the fish. So this is actually something, again, important to take into account when you're looking at different dependent markers. So now we had some idea about potential markers we could look at and different methods we could use. So we wanted to apply an intervention. So we asked whether age-related neurobiological changes in the aging zebrafish brain vary with dietary restriction and or it's mimetic. So these two studies were done both by Aicha, pushed one forward, and Dylan, my current PhD student, also worked on one. So for this, in both cases, we applied, again, a caloric restriction. However, we did a little differently this time. Instead of a daily reduction in the total caloric intake, we actually did what's called an intermittent fasting, or every other day feeding. So in this case, we actually, to limit the total number of calories that fish is taking, we actually fed them on a schedule. So they were fed one day, fasted one day, fed one day, fasted one day. And this method has also been used in rodents to induce a daily, what amounts to a daily reduction in caloric intake, or similar to the daily reduction in caloric intake. And what they've shown in rodents is that the neurobiological consequences and the health span and lifespan consequences are not different between animals that have a daily reduction versus those that have this every other day feeding. The other thing, though, what was important is this was actually one of the first I think the first study is to utilize a true caloric restriction in fish, because the only other one that had been done was actually 6 weeks of not okay, I'm coming down to the last part. No, of basically starving animals. So for this, so for this study, we actually had different schedules of feeding. So zebrafish not only get dry food, but they also get artemia. So artemia are actually live shrimps. And these live shrimps are not only a form of nutrient, but they're actually a form of environmental enrichment. So in the caloric restricted animals, we didn't actually want to not give them this form of environmental enrichment. So animals that were ad libitum fed basically had more food, even though it's limited to a certain extent. There was actually food remnants left. Whereas caloric restricted animals actually had every other day feeding, where they had no feeding and then certain food. So they had an intermittent fasting. If you look at the body weight, and by the way, we did this for 12 weeks. We did it in young animals, and we also did it in old animals. So what you'll notice is that dietary restricted young and old actually have significantly less body weight compared to their AL counterparts. And this body weight actually these changes in body weight didn't actually prevent an age-related increase between the dietary groups. So fish continue to grow even into adulthood, and they actually, so the short-term dietary restrictions didn't change that. Then in Aicha's study, she took the brains out after she had labeled the newly dividing cells with BRDU. She did immunostaining and scanned them on a confocal. She counted the number of newly born neurons and then normalized it to the area of the pallium. So in case there were changes in the brain size, that wouldn't actually affect the total neuron number. When she did this, she actually showed that, similar to another study, there are age-related declines in these newly born neurons. And interestingly, in our study, in the dietary groups, dietary restriction didn't change these number of newly proliferating neurons. She also looked at a age-related marker that is altered, a cell senescent marker, SA beta galactosidase, which is increased in different tissues with aging, and showed that its levels didn't change with age nor diet. So they actually increased in both AL and dietary restricted animals, but no effective diet. And then interestingly, she also looked at telomere links, which are reflecting the proliferative status, or reflect the proliferative status of the cells. And what she saw is that with diet, there are significant decreases, both in young and old. With aging, we actually see a decrease in the total telomeric link. But what dietary restriction is doing, even short term, is actually lowering and stabilizing this change into age. So in a similar manner that I've talked to you about with this stabilizing trend that dietary restriction does, it also appears to be, even in a short-term model, changing telomere link. So for the study that Dylan did, she actually did a dietary restriction where she broke it down into 4 weeks, 6 weeks, and 8 weeks. And then she applied the mimetic of dietary restriction, rapamycin, which is actually blocking Mtor activity. So one of the mechanisms through which dietary restriction is thought to be working is through this Mtor signaling pathway. And you can see that basically the idea is that insulin-like growth factor 1 is thought to increase the activity of this Mtor, mammalian target of rapamycin. And so in this case, dietary restriction is actually affecting Mtor activity by potentially blocking it. And if you use a mimetic of dietary restriction, which is rapamycin, it's thought to be doing the same thing. So for these studies, she actually took young and old fish. She divided them into a dietary restriction group, a rapamycin-treated group, and an ad libitum fed group. And we kept them in individual tanks with the appropriate number of fish to maintain a social hierarchy. She looked at the activity of Mtor because, again, that's a target of both this drug and dietary restriction. And what we saw is that in young animals, dietary restriction and rapamycin, blocker of Mtor, decrease Mtor activity. This Mtor activity doesn't change with age. Also, IGF-1 levels decreased in the young rapamycin-treated animals, but didn't change in old. So this, because we're applying a short-term intervention here, the short-term may not be strong enough to get similar effects that are happening in old animals. Then she looked at different cellular and synaptic markers. So this proliferative marker, it increased in both young and old dietary restricted with no change in rapamycin. Synaptophysin, the presynaptic marker, was higher in old animals compared to young, and also increased across from the CR mimetic groups. In terms of the postsynaptic density protein, there was only a change seen between young and old pharmacologically treated animals. And in terms of the inhibitory synaptic protein levels, Geffen actually, I'm just going to summarize just 1 second and then I'll get to the end. So basically, with inhibitory animals, they are decreasing in old animals with our dietary versus rapamycin treatment. I had wanted to talk about an overfeeding regimen, but I'm not going to do that in the interest of time. So just to conclude, these data are showing that brain aging is sexually dimorphic in zebrafish, and it's similar that what we see in humans and rodents. It doesn't result with not resulting in significant neuron loss. The levels of genes and proteins related to cellular and synaptic regulation are differentially expressed in young versus old, as well as male versus female brains. And these changes may result in functional differences in the synapses during aging. And finally, a short-term dietary restriction doesn't alter the course of total cell proliferation. However, cellular and senescence markers vary depending on the age of the animal. This may indicate that some of the beneficial effects of a dietary restriction and mimetic may be done through subtly altering the cell cycle dynamics. And our preliminary work, which I didn't talk about, related to overfeeding, indicates short-term calorie increases are not detrimental to the aging brain. So currently, what are we doing? We're continuing dietary interventions such as caloric restriction, overfeeding, but we're applying high-fat diets as well as employing mimetics of both of these. We're using knockout animals of nutrient signaling pathway to understand direct effects of these interventions on cellular, synaptic structure and function. And future studies are planned on examining functional consequences of these differentially expressed genes in terms of age-related changes in cell synapses as well as cognitive outcome. I just wanted to acknowledge my group. I'm not going to go through every individual, but both my group members, B. Kent, my other collaborations across different universities and alumni. And finally, my funding sources, both for the rodent studies and zebrafish studies, as well as the university support. So thank you very much.
It was wonderful, and we thank you so much. And we would like to ask some questions, but we don't have much time, so we're gonna choose some questions and we're gonna ask you them. What are the other advantages of choosing zebrafish models for brain researches, such as Parkinson's disease research?
So the other advantage of zebrafish is that, one, if you make a genetic manipulation in them, unlike mouse, which gives you, a very small number of offspring, zebrafish will lay 300 eggs. So if you want to understand a larger number of subjects, what's happening in the large number of subjects, they have that advantage. Also, you can actually then use zebrafish almost as an in vivo cell culture model. So cell culture, when you're trying to understand mechanism, if you do it in cell lines, it gives you some information, but you can actually use the zebrafish embryos as a in vivo cell culture model. And you can also make very quick manipulations in a zebrafish compared to a mouse.
And there is another question about zebrafishes. It says, is there a specific reason why zebrafish were selected for experiments?
So they're cost effective. The facility was set up in my university at that time. I had used rats previously. I didn't use mice. And so zebrafish were easy to manipulate in terms of genes. So this is why I actually picked them. And also there's another advantage. Zebrafish actually in some ways are genetically closer to humans than mice. And things like cortisol response, they're very close to humans. So there may be some link that's closer to human than a mouse, but I actually think it's actually important to study zebrafish, mouse, and human together because they actually will answer a lot of questions that just zebrafish to human would be more difficult.
Okay. What is the effect of exercise of neuroplastic?
So what I understand in terms of exercise is that it affects the levels of brain-derived neurotrophic factor and is beneficial so that when subjects are changing into aging or they're aging in their life, exercise will actually keep the levels of this neurotrophic factor high, and that has a beneficial effect on synaptic number and function.
Thank you so much for answering our questions, and thank you so much for the presentation. It was great. It was a huge honor to have you in our Congress, and we would like to present this certificate to show our gratitude.
Okay.
We will share the screen now.
Sorry, I had to speed up there a little bit. I hope you were able to follow it.
No problem. It was great. We were so sorry that we have to cut it, but we don't have much time.
Thank you very much for the certificate. I appreciate the invitation too.
We thank you. It was us quite honored to have you here.
Yeah. Can I ask one question? So in terms of the subjects in the audience, the people listening, are they undergraduates or medical students?
There are all kinds of people here. There are medical students, there are undergraduates, there are engineers, psychiatrists, psychologists. There are lots of kind of people, and there are even younger people here too. So it was real great, and it was a huge crowd here.
Well, I wish everyone well, and I hope everyone stays safe in this pandemic times.
You too, Prof.
I hope we can meet in person at some point, face to face.
Oh, we wish that too. Maybe next year.
Yes, maybe. All right. Thank you very much.